Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your detection of Vibrio cholera, or you can say that finding the Vibrio cholera. See this, what is this Vibrio cholera? We all know that this Vibrio cholera, Vibrio come under the category of microorganisms. That is your bacteria. It is bacteria. And which type of bacteria? It is gram negative bacteria. Okay. It is gram negative bacteria, comma shape, and facultative anaerobes. Why it is important to detect this Vibrio cholerae? If we are doing some experiment, then there is a aim between there is a aim for performing that experiment. So this detection of the detection of the Vibrio cholerae is very important, because when this Vibrio cholerae enter into the human body, primarily it affect the small intestine. Then after that, it produces a toxin which produces the toxin which causes the secretion of large amount of water. Causes the secretion of large amount of water. And when the large amount of water is secreted by the body, then ultimately there is a it will affect the body fluid, and ultimately diarrhea occur, dehydration occur, and sometime it become a life threatening. Okay, due to the dehydration, because all the amount of water are losses in the dehydration, and it will become the life threatening. So that's why it is very important to detect that Vibrio cholerae into our water, especially our drinking water, or you can say that potable water, because this potable water are used in the production of food product. So it is very important to detect this uh, microorganism, that is gram negative bacteria, into the water. Now let's start the lecture. Introduction. Vibrio cholerae is simply a gram negative bacterium. Gram negative bacterium means that when we perform a gram staining, it give a pink color colony. When we see the microscope, there is a pink color bacteria are visualized, and this bacteria are what they are: a gram negative bacteria, curve shaped or comma shaped. It is catalase and oxidase positive, and ferment glucose without gas production. Okay, so they are what they give a positive catalase test, positive oxidase test, and it also ferment the glucose without the production of the gas. Vibrio cholerae occurs naturally in a plankton of fresh, brackish, and salt water. Now, the next is your principle of this experiment. Here we are going to use our two type of the agar, which is just a minute. two types of agar. They are what? They are a selective media, which means that it only grow the Vibrio cholera. So the two type of the agar is what? Bile salt agar and thiosulfate citrate bile salt sucrose agar, which is also known as a TCBS. And it is used as a selectively for the isolation, identification, and enumeration of the Vibrio cholera. So they are what? They are basically this bile salt agar and the thiosulfide citrate bile salt sucrose agar. They are used as a selective for the isolation of this Vibrio cholera, isolation, identification, and enumeration. Vibrio species grow in the presence of relatively high level of bile salt, and it will inhibit the gram positive microorganism. Okay, this Vibrio species grow when the level of the bile salt are high, and when the level of the bile salt are high, they will inhibit the growth of the gram positive microorganisms. And this TCBs are highly active at high pH, that is 8.5 to 9.5, and it will suppress the growth of the intestinal flora other than the Vibrio species. This Vibrio cholera ferment the sucrose, which give a result in a pH shift and the production of yellow colonies. This yellow colonies indicate the Vibrio presence of the Vibrio species. Okay, now the next is about the culture media. See, culture media, we all know that culture media is what? It is a liquid and a semi-solid media which help in the growth of the microorganisms. Okay, so here we are using a different type of media. See, this thiosulfide citrate bile salt sucrose agar and a bile salt agar, they are what? They are specially used for the growth of the Vibrio cholera. Now, first of all, I am going to tell you the composition of all these media. Like the first one is your alkaline peptone. Okay, so basically this alkaline peptone contain peptone, obviously, then sodium chloride. Okay, amount of peptone will be 10 gram and the the sodium chloride, it also 10 gram. Then after mixing it, we maintain its pH. pH will be around 8.4. This is how we prepare a alkaline peptone water. Now the next is your TCBS, that is your thiosulfate citrate bile salt sucrose agar. So it contains agar. Now this composition is for a 1 liter. If you want to prepare it like 250 ml, 300 ml, then you have to calculate the amount of the ingredient according to your need. Now that the ingredient that we required for the preparation of the thiosulfate citrate bile salt sucrose agar, that is your agar, ferric citrate. Agar is required around 14 gram. Ferric citrate is 1 gram. Ox bile around 5 gram. Sodium cholate 3 gram. Sodium thiosulfate 10 gram. Thymol blue 0.04 ml. Bromo thymol blue, sorry, 0.04 gram. Bromothymol blue It is also 0.04 gram. Next is your sodium chloride 10 gram, sucrose 20 gram, mixed peptone 10 gram. Then we required a yeast extract 5 gram and sodium citrate 10 gram. So these composition is again it is for a 1 liter. Now the next is your bile salt agar. It contain this composition is also for a 1 liter and it contain first thing is peptic digest of animal tissue and the amount is 10 gram. Meat extract that is also called a beef extract and the amount that is required are 5 gram. Then we required a sodium chloride 5 gram, sodium tarocholate 5 gram, agar 18 gram and the final pH will be 8.2 plus minus 0.2 and the composition of the nutrient agar was already discussed in our previous lecture. You can go and check it, the composition of the nutrient agar. So this is all the culture media that we have required for the detection of the Vibrio cholerae. Now after that, what we have to do, we have to autoclave it at 121 degrees Celsius, 15 psi pressure for 20 minutes. Okay, then after that, our media is ready for the culturing. Now the next is your procedure. Disinfect the surface of the bottle, pouch, cup which contained a sample with the help of the 70% of the ethanol or 75% of the ethanol, and along with it we have to also clean the laminar air flow with the help of the 75% of the ethanol. Then we have to switch on the UV light for 20 minutes. Okay, then after that we have to start our work. Now thoroughly mix the sample by a vigorous shaking so that we will achieve a uniform distribution. After that we filter the sample with the help of the sterile membrane filter, and the pore size of the sterile membrane filter will be 0.45 micron pore size, and the place the filter in 50 ml of the alkaline peptone water. After filtering the sample with the help of the filter paper, we get a filter. Then this filter is mixed with the 50 ml of the alkaline peptone water and incubate it 37 degrees Celsius for overnight. Now after overnight, we um, take a streak loop fold on PCBS and a BSA medium plate, and then again we incubate it at 37 degrees Celsius for overnight. So what we have to do, we have to prepare a two separate plate. One is your TCBS and one is your BSA. Now the after incubation at alkaline peptone water, we get a like some colonies. So we streak that loop on that media separately, and then we place that plate inside the incubator for overnight at 37 degree Celsius. Now, suspicious colonies of Vibrio cholerae on TCBS appear as opaque yellow color with entire round margin. If there is a Vibrio cholerae present in the sample, then we get what? Opaque yellow colored colony, and these colonies are rounded by a margin. Vibrio cholerae colonies also show a distinctive appearance on a BSA medium. That's why we're using the two type of the media. One is your TCBS and another one is your BSA medium. Then the confirmation test. When we get a colony, then after that we perform a confirmation test. So the first thing is we perform a biochemical confirmation. If there are a typical suspected colonies, now plate out at least 5 selected colonies from each positive agar medium on nutrient agar number 2, and then again incubate it at 36 degrees Celsius plus minus 2. So obviously we place it at 37 degrees Celsius for 24 hours. Then after that we proceed the further biochemical or serological confirmation. Okay, when we get a suspected colonies, now we take a 5 selected colonies, and these 5 selected colonies are inoculated into a agar medium on a nutrient agar number 2 plate, and again incubate it at 36 plus minus 2 degrees Celsius for a 24 hours. After 24 hours, when we again get the colonies, when we perform a biochemical and serological confirmation. The first biochemical confirmation is your gram staining. Obviously, we all know that this Vibrio cholera are what? They are a gram negative bacteria. So I already told you it will give a pink color colony. Now, gram stain, the isolated, the colony, typical Vibrio cholerae are gram negative, curve shape, round when observed under a microscope. Then test for the motility. Nutrient agar for motility is used to check the motility of the Vibrio cholerae. Inoculate the stubbing with a straight wire into the glass tube to a depth of the 5 mm, and then again incubate it at 37 degrees Celsius for 24 hours. Then the motile strain shall be found to show growth on the surface of the medium outside the inner glass tube. Then again travel through a entire medium. If there is a no motility then observed, then we keep it at a room temperature for 4 to 6 days, and after that we found out any evidence of the motility is present. Then we perform a test for a catalyst. It is very easy. We take a streak on a nutrient agar slant and allow to grow for 24 hours at 37 degrees Celsius. Then we take a slide. Now in slide we put a culture. Then after that we put a hydrogen peroxide. Okay, if the catalyst test is positive, then the bubbles are shown and it will indicate that the presence of the catalyst. This is how we perform the catalyst. Then the next is your serological test confirmation. Suspicious growth of the Vibrio cholerae is tested by a slide agglutinating by using a polyvalent cholera typing syrup. This is what? This is a higher titer syrup which have a combination of a Ogawa and a Inaba serotype, and the growth is emulsified in a drop normal saline and the smooth suspicious mix with the drop of the cholera serum, and if there is a positive reaction then it will show a clumps within a 30 minutes. Expression of a result, presence or absence of the cholera species given after the confirmation of the presumptive colony in the sample examined. So it is equal to presence or absence per x ml of the sample filter. So this is how we express the result, and this is how we detect the Vibrio cholerae. This is all about the today's lecture. I hope you will understand, but if you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or for a further query, you can also ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.